Okay, hello everyone and welcome to our webinar tonight. My name is Shelly Korean with SkyPass Foundation and this is our monthly Ask the Doctor series, and our topic for tonight is clinical trials and new research in PD. And I'm honored to introduce you to our guest for tonight, Dr. Tanya Simuni of Northwestern University. She has a wealth of knowledge and experience in this area. Dr. Simuni is the lead investigator of a number of clinical trials on experimental pharmacology, non-motor manifestations, and pharmacological management of PD. Dr. Simuni is currently the investigator of more than a dozen PD clinical trials, and she serves on a number of steering committees for the PD National Clinic Trials, several committees of the Parkinson Study Group, as well as the National Parkinson Foundation. She is also an active member of the American Academy of Neurology, American Neurological Association, and the Movement Disorder Society, as well as the Parkinson's Study Group and REM Behaviors Disorders Study Group. That's a mouthful. Thank you, Dr. Simuni, for being here. We're excited to hear from you. But before we get started, I know that those of you who are regular attendees, you're probably thinking, Shelly, we're used to seeing you on the screen when we get started, and we knew Dr. Simuni would be here, but who is this other person and where is Dr. Chitnis tonight? Dr. Shilpa Chitnis unfortunately couldn't make it. She had a last-minute schedule conflict that she couldn't avoid, but in her place, I'm excited to introduce you guys to Dr. Mazen Elkurd, who has graciously agreed to co-host and conduct our interview for tonight. Dr. Elkurd is actually a former fellow of Dr. Chitnis's, and he's an assistant professor of neurology at UT Southwestern. So he is going to be co-hosting with me and asking Dr. Simuni some questions about our chosen topic for tonight. And before I hand it over to him, just a reminder, if you're joining us live on Zoom, you can use the chat box or the QA box at the bottom of your screen to keep your questions and comments coming throughout the conversation. And the last 15 to 20 minutes or so, we will dedicate to getting those questions addressed. And some of you submitted questions with registration as well, which I have, and we will get those addressed towards the end of our time together. And with that, Dr. Elkurd, I will turn my camera off and hand it over to you.
Thank you so much for that introduction, Shelly. And thank you, Dr. Simuni, for being here with us today. We all appreciate you and look forward to learning from your wealth of knowledge.
Thank you very much, Shelly. Again, thank you for very warm introductions, and Dr. Elkurd, looking forward to the conversation. Really, I purposefully am stating it's a conversation, it shouldn't be a monologue, and obviously we should give plenty of time to the attendees to ask their questions.
Absolutely. So I'm excited to dive into this topic. It's a big one in the world of Parkinson's, and in fact, it's one of the most important questions that patients are always wondering about in terms of what's the latest research and what's the newest thing going on, what's the future in Parkinson's. These are important big questions that everyone is always interested in. To kind of start us off with the discussion, this has been a long road to get to where we are, and so could you fill us in on the background of this idea of neuroprotection and disease modification in Parkinson's, and the history of what's led us to where we are today? Fill us in briefly on that.
So Mazen, you're going directly for the Holy Grail, right? As all of you living with the disease and their significant others, and obviously also, we as physicians are well aware, there is plenty. We are fortunate in Parkinson's disease compared to other neurodegenerative diseases to have quite an armamentarium of pharmacological options for management of a variety of motor manifestations of the disease that are quite effective, specifically in the earlier phases of the disease, but also in more advanced phases of the disease. Where we're still missing, and not for the lack of trying, is what you have referred to neuroprotection, right? So neuroprotection means, in the definition of the term, is that we're able to salvage the cells. right? The preferred term that is being used in the clinical world of experimental therapeutics is disease modification, because still, as of 2021, we don't have a reliable way to measure the number of cells that someone has or someone is deficient on. So the term disease modification means that we hope that the intervention has salvaged the neurons, has salvaged the cells, but what we're really measuring is the clinical epiphenomena, how the patient's symptoms progress, whether intervention has delayed the progression of the specific milestones compared to someone who is getting no therapy or in clinical trials that will be placebo, right? So the good news are that the world of experimental therapeutics is as active as ever. If you look at the output of the clinical trials, truly, they have not been as active output. Speaking of the world of experimental therapeutics and where to look for the literature, I always actually quote the publication from people living with Parkinson's disease. And that the second year publication from Kevin McFarlane as the first author has come out in Journal of Parkinson's Disease. And again, I'm astounded by sophistication of the authors. The three senior authors are all people living with Parkinson's disease. And that is probably the most comprehensive compendium of updates of clinical trials in development, not only in the United States, but across the globe. So now let's get to the essence of the question. So who are the front runners? No winners yet. It certainly is a marathon and not a sprint, right? Over the last 5 years, definitely the focus has been on developing therapeutics to reduce the burden of the abnormal protein that accumulates in the cells of people living with Parkinson's disease. And the name to that protein is alpha-synuclein, and that is the signature of the pathological changes of the disease. For many years before, there was discussion and debate whether that is the trigger of clinical symptoms or whether that is the epiphenomena. Based on multiple data over the number of recent years, the consensus is that it is the trigger and significant contributor. So there is very active focus of research trying to reduce the load of that synuclein, right? And the front-running studies in that domain are the studies using monoclonal antibodies. So basically to harness the protein that already has left the cell and is in the extracellular compartment. And actually for the first time ever, such studies were launched about 4 years ago by two companies, and both of the companies have read out the results of their studies. So that is terrific. The disappointment is that both of the studies, neither of them has been published, but both of them were presented in public domain. Unfortunately, neither of the study has met its pre-specified primary endpoints. So does that mean that approach, monoclonal antibodies, and the companies that I'm talking about is Roche Genentech that ran the Pasadena study, and Biogen that ran the Spark study. And probably some of you have participated in that studies. And before I go further, just in the spirit of full disclosure and appropriate disclosure, I've been an investigator for those studies. I'm also consulting for those companies, and they have consulting agreements with a number of companies that are in disease modification world. So obviously we'll be talking about the information that is available in the public domain. So returning back to the question, negative results of the studies, disappointing, no question about it. Does that mean that the way of harnessing synuclein by using monoclonal antibody infusion therapies is a wrong approach? We cannot say that yet. It is possible because remember what I've said, that those monoclonal antibodies are targeting synuclein that already has left the cell, right? So obviously it originates in the cell, so more proximal, approaching synuclein either at the time when it's forming that abnormal accumulation of the protein, so reducing the production of that abnormal synuclein might have been more effective, but still, it's too early to say whether the studies read negatively because it's a wrong approach, or whether probably the dose was insufficient, or probably the duration of treatment was not long enough, or probably, despite the fact that both therapeutics were tested in newly diagnosed people, still someone who is newly diagnosed has had pathology for a number of years before they develop the first symptom. So probably going even earlier, that before the diagnosis, but when someone develops very subtle features of the disease, what we call prodromal features of the disease, might be the answer to the question. So again, the exciting part is that those programs are developing for the first time in the history of disease modification in Parkinson's. We're targeting the abnormal protein. Concerning, disappointing, negative results, more studies are to come, more data to come, and there are other programs that are targeting synuclein within the cell, aggregation of synuclein that are being launched right now. So long answer to the question, but important question.
So it sounds like this approach whereby you're using antibodies to target that critical protein alpha-synuclein is still in somewhat early stages. There's some promise, although so far the results haven't been as positive as most people would have hoped for this far. But there are some strategies that are being looked into to kind of readjust the way that the studies are done and try again, as it were. Is that kind of where things are essentially with these antibodies?
I believe so. There are other programs that are developing the same mechanistic strategy, obviously different products, different modalities. So we need more data. And we also need more long-term follow-up of those participants. Because while obviously we want to have the data as quickly as possible, right? But we also need to be realistic that the pathology that it has been accumulating over the years, and in some cases over the decades, to truly impact it over 1 year duration of the study probably is unrealistic.
Now, you brought up this really important point, which was whether it's going to be important in the future to be able to diagnose people with Parkinson's even before they develop the cardinal features that we would typically rely on today to make the diagnosis. Is that something that is on the horizon as well in terms of strategies to help improve the diagnostic certainty earlier in the disease to help with these efforts?
So another very important question, huge amount of work is being done. Again, the concept, not to repeat myself, is that Parkinson's does not develop overnight, right? Someone does not wake up with a symptom of Parkinson's disease and yesterday they had normal brain pathology and had no abnormalities in their protein. So obviously it accumulates over the years until the moment the pathology reaches the critical point when someone actually presents with the symptoms. And there is the whole spectrum of what we call prodromal manifestations of the disease, non-motor features, loss of sense of smell, constipation, REM behavior disorder, that abnormal sleep enactment behavior, all of which in multiple studies have been shown to be associated with a higher risk of Parkinson's disease. So obviously you're not going to go after every person who develops constipation in their midlife and say, you will develop Parkinson's disease. We need easily accessible screening tools accompanied by much more specific tools that clearly demonstrate that yes, someone is on the track to development of the disease. I usually joke with my patients that at some point we will have quote unquote colonoscopy as a screening tool for Parkinson's. Hopefully it will be much less painful and much less uncomfortable. But on a serious note, we do have screening tools for diabetes. We do have screening tools for cholesterol levels, for cardiac disease, and so on, right? As of today, we do not have such readily available tool, but a lot of work is being done. And one of the studies that I want to bring to your attention is Michael J. Fox funded Parkinson's Progression Biomarker Initiative study, which is the largest observational study, so no therapeutic drug, but observational study to validate biomarkers of Parkinson's diagnosis and disease progression. So we do have, so again, we're not starting from the clean slate. We do have an imaging tool that probably some of you had or a number of you have heard about, which is called DET scan. What it does, it measures the degree of preservation of or loss of dopaminergic terminals, which do correlate with a number of cells in the area of the brain that is responsible for producing dopamine, the chemical that is largely responsible for the motor signs of the disease. So that scan is commercially available and is being used as a tool to differentiate Parkinsonian symptoms from other symptoms that might be difficult to differentiate clinically. But in the research domain, you actually can do quantitative analysis of the DET scan. And you can say not only this is normal versus abnormal, but you can also measure quantitatively what is the degree of abnormality compared to the healthy age, sex, matched control. The problem is that not everyone who has abnormal, if you start scanning everyone, first of all, that would be exceedingly expensive, right? And besides that, not everyone who has abnormal scan will ultimately develop Parkinson's disease, or it might take a number of years for them to develop the disease. So the approach of PPMI is to take the staged approach. First, start with simple screening questionnaires. Of people who do not have the disease, but ask the questions of the risk factors that we know are associated with the disease. And then to proceed with the further ascertainment. Loss of sense of smell is very commonly reported by people with Parkinson's, and ultimately get to that more sophisticated imaging, biological markers, and so on. So what we aim and what the objective of the study is to develop the panel of biomarkers that can be used. Biomarker is the objective measure, and it can be, again, blood biomarker, ideally something very non-invasive, right? To develop a set of tools, both that could be used for the early diagnosis before someone has diagnosed with Parkinson's disease, or to track the progression of the disease. We're not there yet, but again, truly the level of optimism, extremely interesting data are coming out of us being able to identify that synuclein, not in the pathology of a person who passed number of years later, but actually in the skin tissue, which is not difficult to do a small skin punch biopsy, in the submandibular gland, in the intranasal mucosa. So those are the tissues that we can actually quite non-invasively assess. More work needs to be done, more validation needs to be done. So if nothing else, I really encourage everyone who is attending this webinar and engage your community in participating in those studies. These studies need healthy volunteers. These studies need first-degree relatives of people with Parkinson's disease. These studies need people who carry genetic variants associated with increased risk of Parkinson's disease. So no question about it, similar to Alzheimer's disease, to other diseases, we're trying to move into pre-disease state, so that rather than talk about slowing progression of already diagnosed disease, to start talking about disease prevention. So far, it's the future, but it's the future that is coming.
Absolutely crucial effort to really find ways to be able to diagnose this disease earlier on, and those are all really promising avenues to be able to use one or more of those to be able to do that, and hopefully that may lead to a more successful trial of one of these antibody drugs that could potentially clear out alpha-synuclein and modify the disease in that way. So far, we've talked about those antibody drugs that are being studied, but of course, there are multiple other avenues in terms of disease modification or neuroprotection in Parkinson's disease. So one of those is looking at genetic mutations that are associated with Parkinson's disease, such as GBA and LRRK2 mutations, both of which there's been work on using techniques to modify the activity of those genes in order to help with either the confirmed genetic forms of Parkinson's associated with it, or even the typical or idiopathic Parkinson's disease. So where are we with those types of studies?
Okay, so before I start answering directly the question, let's step back, right? And for the group, and again, I know that this is a highly educated group of people with the disease or their family members, but Parkinson's, whenever you're trying to cure the disease, slow progression of the disease, in order to do that, you need to understand the root of the problem. What is the course of the disease? And we still don't know that. And that's what we tell the patients, right? The two biggest buckets are environmental exposure to kind of very non-specific list, aside from selected specific toxins, right? And genetics. And the last 20 years really have been an explosion of knowledge of genetics of Parkinson's disease. So what does that mean at an individual level? At individual level, overall, still Parkinson's is largely non-genetic disease, right? If you look at the PD population at large, 5 to 10% of the people would carry genetic variants that clearly are known to be associated with increased risk of the disease, right? So why do we care about 5 to 10%? Because these 5 to 10%, not only, obviously, it's important if we have the strategies to prevent or to slow their disease, even if it is for 5 to 10%, it's hugely important. But no less important is that the variants that they carry are linked to the biology that is present in a person with no genetic variant. So it is kind of concentrated test tube, right? In which if we show that the intervention helps, ultimately, potentially, we could apply it to people with what we call sporadic disease, no known mutation. So Dr. Elkurd has called out the two genetic variants that are the highest risk associated with the disease. One of them is what is called autosomal dominant, meaning that it's passed from one generation to the other, and one quote unquote bad gene is enough to manifest the disease. That is LRRK2 variant, and the other one is GBA, which is the gene responsible for the disease known as Gaucher disease, which is the most common lysosomal storage disease. But Gaucher occurs in a person who inherited two quote unquote bad genes, one from the one parent, the other one from the other parent. And it was quite important observation about 20 years ago by clinicians who were taking care of people with Gaucher disease, that either they or their unaffected family members had increased manifestations of Parkinson's disease. So, and what was found out that carriers, meaning they do not develop Gaucher disease, they carry only one bad gene, the other one is healthy, still have higher risk of developing of Parkinson's disease. That is linked to still that lysosomal dysfunction. Lysosome is the part of the cell that basically garbage collector of the cell. It clears bad stuff out of the cells. And obviously, if your garbage disposal is not working, you start accumulating bad stuff and the cells start working not properly. So to fast forward again, in the last 5 years, for the first time ever, there are therapeutics being developed targeting those particular genetic variants and underlying biology of those variants. GBA is in the forefront. The first program that went into GBA therapeutics was Sanofi Genzyme that tested what is called substrate reduction therapy. And there are multiple different ways to approach that GBA mutation. In people with Gaucher disease, you just give them enzyme replacement therapy, and you do it peripherally to improve the symptoms of the bone disease, of the spleen disease, of the liver disease. You cannot do that for the brain because that enzyme just doesn't get in from the blood in to the brain. So Sanofi was the first company that extremely important study that basically said, we'll be able to find those people, right? Because up until recently, we were not doing any routine genetic testing for people with Parkinson's disease. We will be able to find those people. We'll recruit them into the study and we'll test this particular mechanism. So again, the study has been completed. The results of the 2-year intervention were publicly presented. Again, unfortunately, negative results of the study. Does that reduce enthusiasm about targeting GPA? Absolutely not. It is still in the forefront. And as I have said, they tested particular mechanism. There are other mechanisms. Can we modulate the enzyme, not by dripping it into the brain, but something that is called allosteric modulators or chaperones. I'm not going to quiz you on those words, but something that does get into the brain, and oral molecules that do get into the brain and improve the function of GPA that is present there. So indirect modulation of the enzyme. Such programs are in the earlier phases of the development, but again, we anticipate they're definitely already in clinic, and we anticipate that more of such molecules will be tested. So at some point, it's the critical mass, and we are quite optimistic that there will be winners. Similarly, LARC variant is actually the most common genetic mutation associated with increased risk of Parkinson's disease in the overall PD population. Common means it's about 1.5 to 3%. However, in certain ethnic categories, specifically Ashkenazi Jewish population, Mediterranean population, much higher incidence of the LARC mutation carriers in people with Parkinson's disease. And again, a company called Denali is trailblazing and reported results of their early phase 1 study that actually the oral molecule does get into the brain, does improve the function of the kinase that the gene codes for, and now are launching the study in collaboration with Biogen. So again, no winners yet, but a lot of exciting signs, a lot of very sound signs that we anticipate will translate into the clinically meaningful results.
So these genetic mutations that are associated with Parkinson's disease have candidate molecules that are being investigated basically to work on decreasing the activity of those genes in association with the genetic forms of Parkinson's, and that may even in the future translate into non-genetic forms because of the way that these genetic forms of Parkinson's sort of shed light on the underlying biology of even non-genetic forms. Is that accurate?
Yeah, it's accurate. I wouldn't, so again, it depends, modifying the activity, right? Because in LARC state, it's actually increased activity of the kinase. In GBA, there is insufficient amount of the enzyme, so the directionality might be different depending on which gene and which pathway, but modifying and normalizing the activity.
So that's just another potential avenue that's being investigated, again, in somewhat early stages, and in the beginning may be only applicable to a small minority of people with Parkinson's disease, but may open the doors in the future to more therapies for the general population, which is a great thing. So moving on from the genetic therapies, there are multiple other small molecule and other medications that have already been on the market for treatment of other conditions that have been looked into and investigated as to their potential to play a role in treatment of Parkinson's disease. Some of them have shown some results and others not. And so the one that many people may have heard of in the past few years that there was a lot of interest in and some controversy is a molecule called nilotinib, which is a treatment for leukemia. And recently we had some results on that. Can you share with us what was learned through those studies?
Sure, absolutely. So as Dr. Elkurd has absolutely accurately indicated, nilotinib is one of the commercially available drugs for treating of leukemia, and in the lab, not because it treats leukemia, but because it targets particular kinase, c-Abl kinase, was shown to have neuroprotective effect in the animal models. And for that reason was of interest, and a program was launched by one of the institutions in the country, very small cohort looking at the safety and then reporting quite remarkable efficacy. As a consequence of that, there was tremendous level of interest in the Parkinson's community. The same group has launched a larger study as a single center study, and in parallel with that, Michael J. Fox Foundation sponsored a multi-center study that actually I was honored to lead. The objectives of both studies were not to demonstrate efficacy, but to test safety. Obviously, it's a cancer drug, safety, tolerability, ability of the drug to get into the brain, right? Because if it stays in the periphery, it's not going to do much. And as exploratory measures to look, does it change the trajectory of the disease? So both studies have read out, both studies actually were published, both studies had very similar results, that there was acceptable safety. However, in a very carefully selected population. In our study, 40% of otherwise qualified people did not qualify for the study because of some blood work results or something else. So in very carefully selected population, it was reasonably safe, it was tolerable, but it had very poor brain penetration, did not change the biology that we expected it to change. And for that reason, our conclusion of the study was that there is no role for testing nilotinib in SPD modifying intervention. However, we still are enthusiastic about this mechanism, and the drugs that have better brain penetration, higher exposure in the brain potentially could have a place. The other group had this very same results, but some very different conclusion of the study, saying that they are interested to move ahead with a phase 3 study. I don't know where it stands.
Okay, and so there's some potential for at least a similar mechanism to be explored further, if not this particular drug, perhaps.
Right.
What about some of these other molecules that have been used for other drugs? For example, exenatide, which is a diabetes medication. There was some interest in that particular medication for use in Parkinson's. Where are we with that?
So very good question. Again, a lot of activity looking at hypoglycemic drugs in Parkinson's disease, not because people with Parkinson's have elevated blood sugar, right? But because the compounds that specifically belong to glucagon-like peptide inhibitors, and that's what exenatide is, actually have a way to modulate Parkinson's related mechanism at multiple levels. There is accumulating data that there is inflammation associated with the disease, that there is impaired metabolic cascade, that there is mitochondrial dysfunction, and all of that potentially could be targeted by the drugs in that class. Exenatide is the front runner. The group in Great Britain has done a very well-designed phase 2 study in people with moderately advanced disease. It was phase 2 study, so it was not large enough population to really answer the question of efficacy. They did see the beneficial separation between the groups treated with the drug versus placebo, but by the end of the study, when the drug was discontinued, the groups came together. And because the groups came together over a short period of time, the question was raised whether it was truly the separation was due to rescuing the cells, or whether it was some kind of effect on the improvement of the symptoms. Either one is important, right? So the same group has launched now the definitive phase 3 study that has started recruiting. And in parallel with that, another company, a commercial entity, Neurology, has launched a study of chemically similar compound, but it is deuterated exenatide, meaning long-acting exenatide in the phase 2 clinical trials right now. And there are a couple other of GLP-1 compounds in early phase clinical trials in Parkinson's disease. So again, interesting pathway, a number of clinical trials, no readouts yet. One disclaimer: whenever we talk about approved compounds, it doesn't mean that you need to that you should be rushing to your primary care physicians asking to prescribe those drugs. Until the moment we have results of the studies, all of you recognize that every drug is associated with side effects, and I strongly discourage any off-label use until the moment we have sufficient data to support such.
Very important disclaimer. And so continuing on with this sort of same line of looking at these existing drugs, a couple others that are blood pressure medication is trypsin, and then inosine, which is a supplement that's available. So what can you tell us about those two?
Okay. Both were studied in phase 3 studies. Both were based on very solid science, strong preclinical data, epidemiological data supporting that these class of agents reduce the risk of developing Parkinson's disease. Both, unfortunately, had negative readouts of the studies. So again, that's what science is. And I hope that you're not going to leave this discussion with a sense of pessimism and that nothing works, right? What all of us have to recognize that for one molecule approved by FDA for whatever indication, there will be tens and tens that fail, specifically in the world of central nervous system diseases, specifically in the world of degenerative diseases. And as long as we try for the right reason, based on the right science, and as long as we make definitive conclusions, whether there is a chance for success or not, we will be facing failures, but we learn on every single failure.
So absolutely, it sounds like there are multiple different avenues being explored. And while it's true that we haven't arrived at that, as you called it in the beginning, the holy grail of the treatment of Parkinson's disease, there's absolutely no reason for pessimism just yet. There's quite a bit of hope in terms of open pipelines and avenues that are being furiously explored by researchers and drug companies alike. So absolutely open potential there. Now, before we move on from this topic of disease modifying and then spending a little bit of time talking about symptomatic treatments, Parkinson, an important question that many patients ask about and are very interested in is stem cell therapies in Parkinson's disease. This is something that has garnered lots of interest, some controversies. So what can you tell us about those therapies?
You're absolutely right. Not a single conversation with a patient's lecture goes by without a question about stem cells, right? The history of stem cells, or let's precede stem cells with fetal cells, right? The idea is the same. You're coaching young, undifferentiated cells to differentiate into what you want them to produce, right? So fetal cells obviously have a lot of ethical considerations and limitation with the development of the induced pluripotent cells. You avoid the controversy of harvesting the cells from the fetuses to generating them, but the idea is the same, right? You're coaching the cells that you're delivering to the brain to produce something that the brain is deficient on. So the history of transplant therapeutics trying to deliver dopamine producing cells to the brain of people with Parkinson's disease now goes back more than 35 years, and actually started from the trials of adipose tissue transplant that were done originally in Mexico, and then had remarkable results only to in the open label format, only to demonstrate that in the controlled environment, placebo-controlled studies had no benefit whatsoever, followed by a number of studies of fetal dopamine producing cells, and again, negative results in number of groups, different protocols, but negative results at the cohort level with a few selected participants, younger participants, who had actually long-lasting benefit. And because of those negative results, kind of the world came to coalescence and quiet, probably for about 20 years. And over the last couple of years, there has been resurgence of interest now looking at actually induced pluripotent cells, iPS cells generated, and with a better technology. Whether the technology will solve the problem, I am not a pessimist, but I am maintaining my equipoise. And my reasoning for that is that Parkinson's is much more, we can produce the cells that produce dopamine. We can deliver them in the brain, and we can do it safely. And we know that from the previous experiments, right? The problem is that Parkinson's is the disease of much more than just one chemical dysfunction. We talked about dopamine a lot, but that is only one chemical, what we call neurotransmitter, that the brain has reduced capacity to produce. So unless we address the mechanism of the disease in its entirety, I personally am skeptical that we truly will be able to meaningfully change the trajectory of the disease. It probably will be a viable approach to see whether it could be an alternative to deep brain stimulation surgery, basically to produce a long-lasting way of producing dopamine in the brain of the person, right? Or improving production of dopamine. So returning back, everyone asks the question, so when will I get stem cells? There are studies that are starting. There is a study in Australia. There is a study that was announced by the New York Stem Cell Society to be launched. All small protocols, to the best of my knowledge, not a single one is recruiting so far. There are, on the other hand, plenty of freestanding, not scientifically supported shops. I purposefully am calling them shops and not the programs that are advertising stem cell projects. I strongly discourage patients to be participating in these kind of, those are not studies, but it's fee for service, and strongly advise the patient to talk to your physicians and to get regular update on the status of the field.
Absolutely. Well, I definitely want to leave some time for questions from our participants. Real quick before we move on to that, I know that there are some important trials that are not necessarily disease modifying, but still very important in the field of Parkinson treatment. And so what are some of the more important ones that are ongoing in terms of symptomatic treatments for Parkinson's disease that you think are important for our participants to know about today?
Extremely important question, right? Obviously, we should shoot high, right? We should be shooting for the goal of curing the disease, slowing the progression of the disease, but we have to be extremely cognizant that every patient who comes to our clinic wants to have the improvement of their symptoms, right? So we do have effective therapeutics, but we need more. And I believe that the last 10 years, and specifically the last 5 years, if you want to give one title to that, it's really optimization of levodopa therapeutics. Carbidopa, levodopa, what a lot of you know as Cinemet, is the mainstream treatment of Parkinson's disease. It's delivering that deficient dopamine to the body that is being picked up by the brain. It is still the best drug that we have, but it also has a lot of limitations. It's a short-acting drug, and because it's short-acting drug, people have to take it multiple times a day, which leads to uneven response to medications, pulsatile delivery, dyskinesia. So there is a number of programs looking at what we call optimization of levodopa delivery, right? And delivering it continuously. And the programs that are probably the biggest interest are the delivery by under the skin, small needle, akin to the insulin pump. A couple companies are in development. One of them is Neuroderm, which is sub-Q, sub-Q meaning under the skin delivery. The person does need to change the needle and the setup, the cassette, every 24 hours. And another company that the same concept, obviously different drug, is AbbVie. Both of them should be reading in the next, based on their information on clinical trials, in the next 12 months from AbbVie, probably a little bit longer from Neuroderm, and if shown to be effective and well tolerated, these are phase 3 studies, so they look at efficacy compared to the standard levodopa, would definitely be an important addition in our armamentarium of therapeutics for people who have uneven response to medication, so moderately advanced disease. So that's those programs certainly to be of information to people with PD. Definitely an area of tremendous unmet need are symptoms that we currently do not have good therapeutics: balance, falls, cognition. And again, over the last probably 3 years, we're finally seeing pharma stepping into developing therapeutics for those quite debilitating symptoms, and they used to shy away from them because those are difficult, those are tough. So there are a couple companies looking at different classes of drugs, looking at reduction of falls. There are companies that are looking at the cognitive manifestations of the disease. So now we're talking about more advanced spectrum of the disease, but again, there are therapeutics being tested there. So really hope that in the next 3 to 5 years, we'll have positive readouts of those studies.
It's wonderful. So really important studies taking place at every spectrum of the disease, from all the way from preclinical to the most advanced stages. So I'd like to move now to some of the questions that have been posed by our participants, starting with a question that was submitted early with registration. One of the participants asks, how do the clinical trials and new research relate to PSP?
PSP stands for progressive supranuclear palsy. It frequently is referred to Parkinsonism plus or atypical Parkinsonian syndrome. It is triggered by accumulation of a different protein, tau. And again, out of the spectrum of PD conversation, a lot of studies have been running over the last 3 to 5 years, negative readouts of those studies, but again, it doesn't mean that they stopped. The concept is the same. The front-running programs were monoclonal antibodies, but targeting not synuclein, but tau accumulation protein, read out negatively, but again, it doesn't mean that the programs will hold. Different molecules will be tested.
The other question is, have any of the alpha-synuclein targets that failed in PD been tried in other synucleinopathies, such as Lewy body, MSA, etc.?
So none of the programs that I talked about, synuclein targeting therapeutics, the monoclonal antibodies have yet been tried in either multiple system atrophy or Lewy body disease. But other approach to synuclein vaccine. Early phase studies have been done in multiple systems atrophy. That was a safety, a FIRIS program, different approach, basically giving active immunization and allowing the body to develop their own antibodies. The program read out demonstrating safety, tolerability, but for some reason, no further activity has been yet. Lewy body dementia, again, kind of more complicated, more multifaceted spectrum of manifestations. So I suspect that probably we need the first positive readout in Parkinson's disease as clean experiment before they move into the Lewy body disease.
Okay. Here's another one. A participant asks, I read an article about exposure to pesticides being linked to the development of PD. Any new research in this area?
You're absolutely right that when we talked about environmental risk factors, probably at the top of the list are pesticides. It has to do with impact, basically toxic exposure to molecules that impact energy part of the cells, mitochondria. More epidemiological studies in regard to therapeutics that actually gave us deep understanding of the role of mitochondria in pathology of the disease. There are therapeutics that specifically targeting mitochondria, but that's, to the best of my knowledge, the extent of the current therapeutic development.
So one of our other participants asks whether there are any early stories from PD gene.
Okay, so first of all, I can see the name of that participant and very much appreciate your tremendous contribution to the field as active contributor to the design of the studies and serving on the steering committee. Very much appreciated. In regard to PD gene, the study has, so PD gene is the program supported by the Parkinson Foundation, the objective of which is to give the opportunity to people living with Parkinson's disease to have a Parkinson's genetic panel testing. And the objective to empower people to know their genetic status. If they know their genetic status, they might be candidates for the studies, and to see what is the impact on are people interested to be tested, what is the impact on their clinical care, and so on. So the study has crossed 1,500 to the, as I believe I'm saying the number correctly, of people tested, and has published first the design of the study, then the earliest recruitment marks have been in preparation. The data will be presented at the Movement Disorders Congress. Again, this is the study that will not lead to the discovery per se. This is the study that will enable discovery, right? Because we talked about GBA therapeutics, we talked about LARC therapeutics, and if we don't create the mechanism for genetic testing of people with Parkinson's disease, how will we ever recruit participants for those studies? And we also need much more people that we know their genetic status, because those genetic variants can behave differently from the standpoint of their clinical manifestations. Some have milder course of progression, some have actually more rapid course of the progression. So we need that knowledge. Ultimately, we need genetic testing for Parkinson's to be part of the clinical armamentarium covered by the insurances, but we need more data to demonstrate that it makes a difference.
Here's another question that just came in. Attendee asks, what is the purpose of having a skin biopsy?
Very good question. So again, returning back to that colonoscopy, but without colonoscopy, right? Colonoscopy looks for any presence of pathology that is an early indicator of the cancer risk, right? So skin biopsy looks for the presence of synuclein in the skin cells, right? If I ask you the question, what would you rather get, lumbar puncture or the skin punch, right? The tiny skin punch. I suspect that majority of you would say, I would go for the skin punch, right? Or if I asked you if I need to get a piece of your brain, or I can go for the skin. So that's the reason for the skin biopsy, to look for the presence of synuclein.
Mm.
And there are multiple accumulating data that Parkinson's pathology, at least in subset of people, could be actually starting not in the brain, but in the peripheral tissue. So for that reason, if we demonstrate that it actually reliably shows presence of synuclein in people at risk for the disease, that could become the screening test.
Yeah, absolutely. But really, really important emerging tool in the diagnosis of synucleinopathies. So one of our colleagues at UT Southwestern, Dr. Brewer-Mixon, asks, can you talk about about freezing of gait and the possible contributory role of anxiety in this PD symptom?
So unfortunately, people with Parkinson's disease are way too familiar that in more advanced stages of the disease, people can develop that debilitating phenomena that they get stuck. I want to go and I cannot. And that's basically the descriptor of freezing of gait. And then usually it happens just when you get started, when you turn, and if the person is in the open space, they start working much better, right? There are those famous videos of a person with severe freezing of gait who really could not get going, would get on the bike and would freely bicycle, would get off the bike and would freeze. So what does that mean? It's really motor. It's not that the feet do not have strength. It's the motor programming phenomena. And multiple different factors could contribute to that. Definitely anxiety, definitely stress can do that, but it's not anxiety generated phenomena. It can be worsened by anxiety, but it is phenomena very characteristic of Parkinsonian state, not unique for Parkinson's disease, can be present in other Parkinsonian syndromes.
Okay. Well, I think that's all the questions that I see so far. Thank you so much for that incredible wealth of knowledge that you provide us. It's a stunning amount of information that you're able to share with our participants and with myself included. So thank you so much for that. I know I've learned a lot, and I'm sure our participants have learned a great deal as well.
So pleasure and really conversation, and I hope that it was useful for the audience.
Absolutely. Thank you both. Thank you, Dr. Elkurd, for filling in for Dr. Chitnis today. Excellent interview. And thank you again, Dr. Simuni. As Dr. Elkurd said, this was such great information, and I know our attendees enjoyed this webinar. Thank you all for attending and for engaging with us and for your interest in our programs. If there's anything that we can do to help you in any way, please let us know. info@skypassfoundation.org. And before we sign off for tonight, if you can just take just 1 second before you shut down your computers for tonight and head over to our website at skypassfoundation.org and sign up for our next webinar on September 7th. We're covering the topic of deep brain stimulation, another hot topic that I know a lot of you have questions on. So you don't want to miss that, September 7th at 7 p.m. Central. You can head over to our website. Registration is now open, and you can submit your questions on there as well. And with that, we will call it a night. Thank you again, everyone. Thank you, Dr. Simuni and Elkurd. Have a great night.
Thank you.
Bye.